आइए अब हम बात करते हैं कॉलम क्रोमेटोग्राफी के ज़रिए पॉली ए आरएनए की प्योरीफिकेशन के प्रोसेस और प्रोसीजर के हवाले से। जैसा कि नाम से ज़ाहिर होता है, कॉलम क्रोमेटोग्राफी में हमें एक कॉलम की ज़रूरत पेश आएगी। अब उस कॉलम के अंदर जो भी मटेरियल फिल करेंगे टू होल्ड द रिक्वायर्ड मटेरियल एंड देन लेटर ऑन टू प्योरीफाई इट, टू एल्यूट इट, दैट इज़ वेरिएबल। Columns are available commercially in the form of plastic tubes, or if they are not available, then one can use pasture pipettes as a column and can fill those pasture pipettes with the required resin or required material that is going to act as column for chromatography. In this case, we are going to use oligo DT cellulose, which is going to be treated with 0.1 normal sodium hydroxide, and then it is going to be added into the empty column, or it is going to be added in the sterile pasture pipette so that we can prepare that column for purification. अगर हम कॉलम क्रोमेटोग्राफी के हवाले से बात करें, हम इसको दो डिफरेंट स्टेप्स में डिवाइड कर सकते हैं। पहला स्टेप कॉलम प्रिपरेशन का है और दूसरा स्टेप प्यूरिफिकेशन का या क्रोमेटोग्राफी का है। कॉलम प्रिपरेशन में वी टेक द एम्प्टी कॉलम, we fill it with the resin or the required material. In this case is oligo DT cellulose, and then we treat or we wash that column with DEPC treated water. The purpose of this treatment is to make that column free from any kind of potential RNases which can degrade our RNA sample. अगर आप इस तस्वीर में देखें, इस तस्वीर में हमने कॉलम को वॉश किया है DEPC ट्रीटेड वाटर से, 3 times वॉशिंग की है, और उसमें से जो फ्लो थ्रू है दैट इज़ गोइंग टू बी कलेक्टेड इन अ कलेक्टिंग ट्यूब, एंड दैट इज़ नॉट रिक्वायर्ड इज़ गोइंग टू बी द वेस्ट. In the subsequent step, we are going to prepare the column for loading, and for that purpose we are going to add 1x column loading buffer. और इस कॉलम लोडिंग बफर को हम कलेक्ट करेंगे कलेक्शन ट्यूब में, और ये प्रोसेस उस वक्त तक कंटिन्यू रहेगा जब तक फ्लो थ्रू की pH 8 या इससे कम नहीं हो जाती. इस प्रोसेस के लिए, इस pH डिटेक्शन के लिए वी कैन यूज़ द pH पेपर टू मेजर द pH, and we will keep on passing this column loading buffer unless until the pH goes below 8. Now this time the next step is going to be the preparation of the RNA sample, which we will load on this prepared column. For that purpose we are going to take the RNA which we extracted from the potential sample, which could be the tissue, which could be the cell extract or any source of the RNA. Then that total RNA is going to be dissolved in autoclaved double distilled water, is going to be heated at 65 degrees centigrade for 5 minutes. The purpose for this heating could be the reason that the intra-strand base pairing should be avoided, or you can say the secondary structure of RNA should be removed. After 5 minutes of heating, immediate cooling down to the room temperature is required so that the secondary structure should not form again, and we are going to add 1 volume of 2 times column loading buffer. यहाँ पे एक चीज़ मैं बताता चलूँ, हमने इससे पहले वाले स्टेप में कॉलम लोडिंग बफर यूज़ किया था कॉलम की वॉशिंग के लिए और कॉलम प्रिपरेशन के लिए. दैट वाज अ डाइल्यूटेड सॉल्यूशन 1x कॉलम लोडिंग बफर. टू प्रिपेयर द RNA sample we are going to use the concentrated column loading buffer, that is 2x column loading buffer. So once the RNA sample is ready to be purified or to separate the poly A RNA, then we are going to add or load this sample on our previously prepared column. जैसे कि इस तस्वीर में देखा जा सकता है, हमारे पास कॉलम है that has the resin or that has the oligo DT cellulose. We are going to add the RNA sample or RNA solution over this column. We have a collection tube, and then we proceed further to purify the RNA. Once the RNA solution is entered or is absorbed in the oligo DT cellulose, then we are going to add 1 volume of column loading buffer. That 1 volume of column loading buffer is going through the column and is going to be collected in the collection tube. This collection tube now contains the flow through, and this flow through is going to be used to purify the poly-A RNA. Once this flow through is collected, we are going to take this flow through again for a heating step, 5 minutes at 65 degrees centigrade, and is going to repeat the same process. That means we are going to load this flow through on the same column once again for collection or for elution of the poly-A RNA. Now once we have loaded this sample, the flow through from the previous step on the column, we are going to add the column loading buffer, the 1x column loading buffer in this column again, and again we are supposed to collect the flow through from this column. Now at this stage, the volume of flow through is going to be more. We are going to perform this step using 5 to 10 column volumes, or if I elaborate it more clearly, 5 to 10 milliliter. And in the collection tubes, we are going to collect 1 volume in each of the collection tube. That means 1 milliliter. So we are going to end up with a series of tubes containing 1 milliliter of the flow through or 1 milliliter of the solution containing the RNA. Using these different elutions, we are going to determine the RNA concentration using the quartz cuvette at 260 nanometer, and we are going to select those tubes which have best concentration of the RNA determined by the spectrophotometry, and this concentration is going to be determined against 1x column loading buffer as blank. These fractions, we are going to precipitate these fractions by the addition of 2.5 volumes of ethanol. Once the RNA is precipitated, then we are going to use the same RNA to collect the poly-A RNA using the oligo DT column again. We are going to use the column again, and we are going to load these eluted RNA samples on the column using 1x column loading buffer, or using elution buffer. Now using elution buffer, we are going to elute the samples, and we are going to determine the concentration of the RNA once again at 260 nanometer. And once we select the best possible concentrations samples of RNA, we are going to pool them, and that sample or that volume contains the best RNA quality and quantity that can be used further. Still, this RNA is not 100% pure. We can have some optional steps to purify the poly-A RNA from this particular purified sample.